All right. So the world is a challenging place, and ocean acidification is a growing problem in today's ocean. It's estimated that about 25% of the CO2 emitted from human activities since the Industrial Revolution has since diffused into the ocean. Now, the ocean acts naturally as a carbon sink. However, in excess, the CO2 diffused in the ocean results in hypercapnic seawater conditions. And this leads to an increased dissociation of hydrogen ions, causing low pH, and also a decrease in carbonate ions and calcium carbonate mineral saturation state, all of which are negative for calcifying organisms. So many of you have heard this before, and this is the hand that we're dealt today in the age of our Anthropocene. Now, what if I was to tell you that not all stress is all that bad, and perhaps stress in moderate doses may even be beneficial? So this is the premise of my dissertation, stress conditioning. And stress conditioning is a priming to an early life experience of stress at a moderate or sublethal level to enhance the ability to cope with stress later in life. You may have heard of stress conditioning referred to as hardening or hormesis. Now I'll explain this briefly about what I mean here. So this is a little theoretical model here. On the x-axis, we have the level of exposure. On the y-axis is the fitness and the performance of the organism. And this horizontal line is a theoretical control individual that's unaffected by any level of stress. In reality, animals experience a linear negative response in which increasing stress results in decreased performance. However, low amounts of stress may be beneficial or have no effect until reaching a threshold in which they induce harm. Stress conditioning is this phenomenon in which that prior exposure allows the animal to cope better during a subsequent encounter. And this has been shown as a fairly conserved phenomenon, ranging from taxa of nematodes to small mammals, and there are a variety of stress types. And this can yield lifespan extension. This can increase cytoprotection or cellular defenses against stress, and also result in thermotolerance, these kind of concepts that we've heard of before. So in light of these changes in our age of the Anthropocene, the growing challenges today, can stress conditioning potentially improve resilience, improve performance of these organisms? So ocean acidification, again, is actually exacerbated in coastal systems, such as temperate estuaries. I bring this up specifically because the aquaculture industry is a hub within these coastal systems. And many aquaculture facilities are for sustainable production of calcifying organisms, such as shellfish. In recent years, ocean acidification has caused a bottleneck for production, in which these early stages are highly susceptible to the low pH conditions of ocean acidification, resulting in harm. Here's an example here. The Pacific Northwest states specifically have dubbed this as a seed stock crisis due to decreased survival and shell malformations of their larvae. This example of Pacific oysters about 4 days post-fertilization. So my question is, can we use stress conditioning, use this concept to potentially improve production and improve resilience in these organisms? This has been shown with oysters through cross-generational experiments and also within generation, looking at carryover effects, for benefits of previous exposure, enhancing the ability to cope with the subsequent exposure, and have ranged for a variety of responses, from increasing developmental rates, increasing survival, and also altering gene expression to cope with that stress. So in my research, I'm going to start with a few questions. I want to know whether this is transferable to another economically important species of bivalve. What is the optimal timing and stress intensity to try and test stress conditioning? And what are the mechanisms underlying this conditioning response? So as for this first question, as you saw from my title slide, I study the geoduck clan. And Pacific geoduck are native in the Pacific Northwest. They're also of very high cultural and economic importance for sustainable production of seafood. And specifically, Washington State is one of the major producers of geoduck. So now we have a target species in mind. What is the optimal stress intensity and life stage to try and test the stress conditioning effect? So consider this cartoon of the life history of geoduck. The adult stages are sessile. They're burrowed very deep in the sediment, and they broadcast spawn their gametes for early larval development in the water column. The reason why I mention this is that there's this transition from a free-swimming larvae to an increasingly sessile life stage, and they begin burrowing at about 30 days post-fertilization. This corresponds with a potential increase in their exposure to low pH conditions. So perhaps these stages may be more tolerant of low pH than these early larval development in the water column. We did perform some pilot tests for stress conditioning where we exposed these early life stages to low pH at the hatchery. Here's showing a conical where we did this research at the hatchery. And we actually found prevalent mortality in these life stages when we tried to test stress conditioning. which isn't surprising considering that cartoon earlier, these develop in the open water column, perhaps less likely to face this low pH condition that's confined in benthic areas. So now for the intensity. Here's a blow-up map of the hatchery where all this research was performed. This is adjacent to the Hood Canal of the Puget Sound in Washington State. And here's a transect showing pH and aragonite saturation. This transect was from August 2008. On the far left is the Pacific Ocean, and this goes into the Puget to the Hood Canal area on the far right. And I have arrows here indicating that at depth, we see a pH less than 7.4 and aragonite saturation state less than 0.5. So what does this tell us? This tells us that this is a potentially relevant condition, not only to this life stage when these larvae sink to the benthic areas for increasing the sessile lifestyle, but also adjacent to the hatchery as a relevant condition and relevant to the native range of the geoduck since they naturally occur in the Hood Canal too. Okay, so now we have our species in mind, the stress intensity and the life stage. My main question of my dissertation is what are mechanisms underlying this conditioning response? So I'll start with my first chapter. My first chapter, we sought to conduct an experiment in which we repeatedly expose these animals to the stress to understand how they respond to the repeat stress under later in life. So my hypothesis is that repeated stress exposure can enhance performance in the geoduck, looking at the response to repeated stress and the carryover effects of this stress. So again, this is just reiterating my questions, but look a little bit more deep here with the physiology. We looked at specifically metabolic rate and shell length as our metrics for physiology. And our question is how they respond to repeated stress with these physiological metrics and all the carryover effects of these previous encounters later in life. So for this research, again, we did all of our research out of hatchery setting, and we focused here on juveniles, about 5 months post-fertilization. And we isolated the juveniles from standard aquaculture practice. So these juveniles were reared at the hatchery, and we isolated them for the experiment. We first exposed them to an ambient seawater condition and also an elevated PCO2 seawater condition, or hypercapnic seawater, to induce a low pH condition. And here's just a little bit of our rig where we use these heat trays as downwellers, sort of a small scale level type rearing practice that is used at a large scale at the hatcheries. So again, a more schematic view here, we started with this initial exposure for 10 days to 2 treatments, the ambient seawater at the hatchery and the elevated PCO2 condition that we completed by pumping in CO2 to the ambient seawater. And here we target a pH, as shown before, that intensity level of 7.4. After that 10 days, the animals were returned for an ambient recovery period for 2 weeks before a second exposure when they're split for the 2 conditions again. So now resulting in 4 treatments here, and the same stress condition, the same elevated condition. All the rest of the animals remaining from this experiment were reared at the hatchery under ambient conditions for grow out to assess the carryover effects of this initial repeated encounters at 5 months post-stress. So here's just condensing this into 2 phases where we have a repeated exposure phase and we have our long-term carryover phase. And just showing the treatments throughout the experiment where we started with 2 treatments, split into 4. So at the end of the experiment, we have the effect of the initial and the second encounter, resulting in 4 treatments. Now what we measured was we measured physiological metrics for performance. We measured them at the start and end of each treatment transition and every other day during the exposures. We measured physiological metrics for performance at the end of the ambient growth period for 1 measurement. What we measured was respiration rate and shell length. Respiration rate we measured as oxygen consumption in 4 milliliter vials, and this 24-well system as you see here, corrected for filtered seawater. And the same animals that we use for respiration rate, we also use to assess shell length with images and side scan. So now for our results. So this is the results of initial exposure. On the x-axis here, we have time, and the y-axis will be the physiological metric here, respiration rate. I will be showing all the figures of this first chapter as these box plots similar to here, where the white box plot indicates the animals exposed to the ambient, and the gray box plot are the animals that were exposed to the stress. So the effect that we found here was an impact of the stress exposure, in which the animals that were exposed to the elevated PCO2 or low pH condition showed a suppressed metabolic rate. Oh, man. Now, as for shell length, we found no effect of shell length under this initial exposure. So to summarize this initial encounter, we see a suppressed metabolic state, but no effect on shell length or shell growth. Now for what happens under the second encounter, where we split these animals to see the effect of the initial and also the effect of the second. So after the ambient recovery period, we found continuous metabolic suppression here on day 0 before they're exposed to the second exposure. So we see a carryover effect of that decreased respiration rate into the second encounter. Now here we have 4 box plots here representing both initial and second exposure. Again, time on the x-axis. The far left here are animals exposed to ambient throughout the experiment, and the far right are animals that were exposed to elevated on the initial and second. And what we found during the second exposure is potential for metabolic recovery, where we did not see an effect of treatment on respiration rate. Similarly for shell length, after the ambient recovery period, we did not see an effect of stress on shell length as we found with the initial exposure. However, upon exposure to the second encounter, we saw a carryover effect of the initial stress, resulting in 4% smaller individuals. So the animals that were first exposed to the stress showed smaller shell length during this second encounter. So we see here evidence of the repeat encounters resulting in metabolic recovery from that suppressed state. And we found that repeated stress affected the shell growth. Now, what about this ambient grow-out period? Remember, we returned all these animals to ambient hatchery conditions for a 5-month period to see if there's any carryover effects of this repeated encounter. So here, instead of time on the x-axis, it's treatment, because we only measured this once, where we have basically the initial time to second treatment. Ambient on the far left, and elevated on the far right for the animals that are repeatedly exposed. What we found here, starting with respiration rate, that animals that were exposed to the second stress treatment had 50% increase in their respiration rate carried over from that initial encounter. As for shell length, we found animals that were exposed to the initial treatment, stress treatment, were about 6% larger. So to summarize our question for this initial chapter, we wanted to measure physiology through respiration rate and shell length to see how they respond to repeated stress. We wanted to see whether this initial stress results in a carryover effect later in life. We found that the repeat encounter resulted in decreased shell length and metabolic suppression. However, after 5 months of ambient conditioning and under ambient for stress-free conditions, we found that stress history may precede growth compensation and recovery of metabolic state. So to summarize what we have so far, we did the pilot test. We tested how did these animals respond to low pH during these early, potentially more susceptible life stages. We found prevalent mortality. So we focused our efforts on the post-settlement stages that may be more tolerant to low pH in their natural environment. And when we targeted the juveniles for this initial exposure, we found that this induced a phenotypic effect, especially during the carryover stage, where we see a phenotypic response and we see carry out during a potential grow-out setting. So for our next step in this research, we want to determine the underlying mechanisms of this conditioning response, try and target additional metrics to determine some mechanisms here at play. So for chapter 2, my hypothesis is that an earlier conditioning period may enable these animals to cope with repeated encounters. If you see here, there's a little bit more added on. We're looking at oxidative stress and cellular stress response to expand upon a mechanistic understanding of stress conditioning. So what is oxidative stress? Oxidative stress is a fairly conserved response to a variety of stressors, ranging from dietary restriction, thermal stress, low oxygen availability. Many different stressors can cause oxidative stress. And it is oxidative stress is caused by overproduction of free radicals, a reactive oxygen species, or when the proteins that function in their removal are compromised. And why look at oxidative stress in this framework of stress conditioning? Well, stress conditioning is sort of can be interpreted as an umbrella term for sublethal stress, enabling organisms to have a beneficial response later in life. And oxidative stress plays a big role in this, with several theories that refer to oxidative stress in this stimulatory phenomenon, such as free radical theory of aging, where early life exposure to reactive oxygen species increases your lifespan extension, and preparation for oxidative stress, where the signaling from reactive oxygen species can increase your response to cellular damage, so you readily respond to for homeostasis and stress. But the trend here is that oxidative stress can trigger cellular defenses. So what about these cellular defenses? There's a conserved stress response to oxidative stress involving a variety of protein families, ranging from signaling to controlling apoptosis to repairing your damaged DNA and lipids. And specifically, I'm interested here in reactive oxygen species scavenging proteins or antioxidants. There's a growing body of literature stating that bivalves upregulate the activity or the expression of antioxidants under exposure to ocean acidification, and that species their baseline activity or expression of antioxidants can reflect species-specific tolerance to ocean acidification. So antioxidants are a good player to look at cellular stress response. So my question in chapter 2 is, does stress conditioning during earlier life stage, you're looking at pedovelger immediate post-settlement, this pedovelger juvenile stage, does this affect physiology and oxidative status when exposed to repeated encounters? So here is a cartoon to summarize what our experiment will look like in the next slides, where we conditioned this earlier life stage to yield pools of both conditioned and naive animals to see how they respond to repeated stress. And this is sort of starting my schematic about what we did here. This is what the pedovelger stage looks like in the center, 30 days after fertilization, when the animals settle out of the water column and begin burrowing. So you can see their extended foot here when they begin burrowing. We expose these animals to 2 treatments, an ambient and elevated PCO2, or the ambient and low pH, for about 3 months to yield pools of juveniles, both naive and conditioned. This is what the juveniles look like at about 5 to 8 millimeter shell length. So looking at this in terms of what the schematic will look like in the slides, we conditioned the pedovelger stage from standard aquaculture protocol. Then we isolated them from the hatchery and exposed them to the elevated PCO2 or low pH condition to yield conditioned juveniles, and then to the ambient seawater to yield naive juveniles, so animals that are not exposed, not preconditioned. And there's our concentrations here, pH 7.4 for the stress. After we have these juveniles in hand, we have this pool of juveniles that are conditioned and naive. We started doing a repeat exposure challenge. But this time, adding an additional layer here for a severe stress. So we expose them to 3 conditions: ambient, moderate, and severe. Here's our pH for these conditions. All these treatments will return to ambient for another 7 days to recover from that stress. Before a third exposure, where we split all the treatments here, all the replicates here, again for an ambient and moderate exposure. So to summarize this experiment, we have the conditioning period where we yielded the 2 pools of juveniles, the conditioned and the naive animals, and then we had the repeat exposure challenge. And here are the treatments to help guide your eye. Additionally, just to help guide your eye again, I'll be showing the schematic when I'm explaining the data here. This gold line I highlight here are the animals that were exposed to the most stress of the experiment. So they were conditioned, exposed to severe, and then exposed to moderate. And here are the animals that were exposed to ambient throughout the experiment. So they were conditioned under ambient, yield naive juveniles, exposed to ambient, and exposed to ambient. So just to help guide your eye, because I'll be showing this schematic in the next slides. So what we measured in this experiment was our respiration rate and shell length for physiology. We measured it every other day at the start of every treatment transition. We also measured tissue biomass and antioxidant capacity from samples at the end of the second and the end of the third exposure. So now for our results of this chapter 2 experiment. I'll be showing the schematic on the left and then the data on the right to help guide your eye about where we are in the experiment, because it's kind of a lot. The post-hoc effects that I'll be showing will be highlighted as a gold print box for the significant post-hoc effects. So first, we had a significant effect of the stress treatments during the second exposure. So I highlight here, showing the second exposure. And here's our data. All the data of this chapter I'll be showing similar to this, but in box plots. Here as the second exposure, the x-axis has ambient, moderate, and severe. And this figure is split in 2 to show the color-coded geoduck as naive and conditioned. Here we have found an interaction between the primary exposure, the conditioning exposure, and the second encounter, where we found animals that were conditioned to the stress had higher respiration rate when faced with that moderate second stress encounter than the animals that were naive. We also found an effect on respiration rate during the third exposure. So here I highlight third exposure, looking at this part of the schematic. Here we have the figure for showing third exposure. Similarly, now on the x-axis here, we have ambient and moderate for the third encounter. The gray scale still refers to ambient, moderate, and severe of the second exposure. And I have it split here to indicate the primary exposure, as indicated by the color-coded geoduck. Here we found an interaction between the primary and second exposures, and the post-hoc differences were specifically for the conditioned animals, where the animals that were exposed to severe during the second encounter had a carryover effect of higher respiration rate than the animals that were encountered in the ambient second encounter. So let's summarize what we see here for respiration rate. We basically see that stress conditioning and repeated stress exposures increased respiration rate. Now for shell size, we found an effect during the third encounter, but this was an interaction between all treatments in which animals that were conditioned, exposed to the severe, and then exposed to the moderate were actually larger than other treatments. As for organic biomass, we found an effect at the end of the experiment, at the end of third exposure, and this effect was of primary treatment, in which the animals that were conditioned were larger than the animals that were naive, regardless of the subsequent stress encounters. So we found here that conditioning and repeat exposure to stress can increase size. Now, lastly, for total antioxidant capacity, we found an effect during the third encounter at the end of the experiment, and this was also an effect of the initial conditioning period, where animals that were naive to stress had higher antioxidant capacity at the end of the experiment, regardless of their subsequent encounters. So here we see that stress conditioning can reduce total antioxidant capacity. So to summarize this chapter, we want to see whether the petavelogid to juvenile conditioning during that life developmental stage can affect physiology and oxidative status under repeated stress encounters. What we found here was a distinct phenotype in which, after the conditioning period, we found animals not conditioned, that were naive, that we could not condition to stress, were smaller and attributed with higher antioxidant capacity than the animals that were conditioned, which grew larger, had a larger shell length, higher respiration rate, and increased tissue biomass under the repeated exposures, and also had lower antioxidant capacity at the end of the experiment. This tells us that there's potential plasticity of bioenergetic and subcellular responses to repeated stress encounters if conditioned prior, and that this post-settlement life stage, as mentioned earlier, may be an optimal timing to elicit phenotypic variation, beneficial phenotypic variation. So now we have this phenotype in hand, and I want to expand upon this further. We have a little bit of a mechanistic understanding of this as expanding upon physiology by looking at stress response and a proxy for oxidative status. And in this third chapter, I was interested in another link to this mechanism, looking at the molecular level. So in my third chapter, it's the same experiment as the second chapter, but we used samples along the experimental timeline for sequencing to look at gene expression patterns. And my hypothesis here is that the early acclimation may generate distinct functions and pathways under repeated stress. So consider a couple terms that we may expect in response to acclimation and repeated encounters. Front loading is one term here, which is an increased expression of stress-related proteins that may enable organisms to cope with episodic stress encounters. There's also front loading, in which animals that are previously conditioned may upregulate only a few important genes under a subsequent stress. So maybe rapidly responsive and also allocating energy to only do a few important genes during the subsequent encounter. So considering these two terms in our conditioning framework, the front loading response may be a prepared response, in which the animals that were initially not conditioned or conditioned may have a more prepared gene expression. Whereas fine-tuned, this earlier conditioning period might allow for the animals to rapidly respond during environmental change. So here's our experimental framework, the same thing I showed for chapter 2. It was the same experiment, and I'm going to run through when we took samples for sequencing. We sampled animals at the end of the primary exposure. So remember, we conditioned the animals for about 3 months to 2 conditions to yield the naive and conditioned animals. And we took samples for sequencing before they were exposed to repeated stress. Next, we have the repeat encounters, where we measured animals at the end of the second exposure, at the end of the ambient recovery, and then at the end of the third exposure. And we measured one animal from each biological replicate that we had for the whole experiment. And this totaled quite a few animals, 141 animals for sequencing. This was possible using TAGSeq, which is a cost-effective alternative to RNA sequencing that allows us to boost the amount of animals that we can measure for sequencing. So how did I measure this data with these repeated exposures? I decided to use weighted gene correlation network analysis, or WGCNA. WGCNA is a tool for finding clusters or modules of highly correlated genes to assess how these groups of genes relate to one another and how they respond to external sample traits. And in this case, my external sample traits are treatment and compounding treatment over time. So I show this in this dendrogram that you can get from WGCNA, where you have these obvious clusters of genes that have similar expression pattern, and you can make figures of these of the mean expression pattern in response to treatment, see what's the trajectory here, what's the directionality of expression in response to treatments. And I consider this as the optimal tool for our experiment, considering that we have these repeat exposures in this complex experimental design. So a little bit about the workflow with WGCNA that I did here. You can output it in that heat map that shows you the significant modules. Again, modules are genes that are highly correlated with one another and grouped because of that high correlation. And you can see how they respond to treatment here, where my treatment is on the x-axis. So on the heat map, where you see that there's values shown, that's showing the directionality of their expression and whether it's significantly correlated and related to treatment. So in summary, you can basically take these modules that are showing a relationship to your treatment and see what their mean expression level looks like regarding your treatment. And once you have these modules in hand, these are groups of genes that have similar expression pattern. You can follow this up with GoSeq and KEGG to look at distinct functions and pathways associated with that gene expression pattern. So to explain this further, I'm going to go over day 0 data to help guide you with this. So we did see one module here. We had 6 modules that showed up for day 0. We were sequenced 8 animals at the end of the primary exposure period. And of these 8 modules, these groups of genes, we found one group of genes was significantly related to our treatment. And here we see a positive correlation with the naive animals, in which this module, these around 3,000 genes, were associated with higher expression in the naive animals than the conditioned animals. We followed up with this module by looking at distinct pathways associated with these genes. And here's our KEGG analysis. I'm going to show all the KEGG pathway analysis as rich factor plots, where on the x-axis represents the number of genes in the enriched pathway proportional to the total genes possible in that pathway. But in summary, the y-axis is going to show significant terms that showed up through pathway analysis. What we found here is that the naive animals, before they're exposed to repeated stress, were exhibiting pathways associated with carbohydrate metabolism, signaling, and biosynthesis. Now, what about during the repeat exposures? So again, we measured animals at the end of the 2nd exposure, end of the ambient recovery, at the end of the 3rd exposure. And interestingly, through WGCNA, we did find an effect of the primary exposure treatment in opposing directions throughout the experiment. And I'll be walking through day 7 to show you what that means. So here's our heat map for day 7, where we found what I'm calling a naive effect module, which is a group of genes that are showing higher expression level in the naive animals. And just to guide your eye, the white geoduck are indicating the naive animals, and the gray geoduck are the conditioned animals. And you basically can see that they have a higher expression level in this group of genes. And I'm calling these naive modules. Next, we have conditioned modules, which are genes that are showing this trend of higher expression in the conditioned animals. And as I mentioned earlier, this was what we found throughout the experiment. So here's showing day 14 into day 21, day 14 being after the ambient recovery, day 21 at the end of the experiment, where on the left we have naive modules, so groups of genes that are highly correlated with one another, and showing this trend of higher expression in naive animals than the conditioned animals. As the opposite effect, we have the conditioning effect modules, where we have conditioned animals showing higher gene expression of these genes than the naive animals. So now we have our modules in hand, our groups of genes in hand. Let's follow this up with seeing what are the distinct functions and pathways associated with these genes. And again, we did this with gene ontology enrichment analysis and pathway enrichment. So to help guide your eye by interpreting this data, I'm going to use this Venn diagram here, color-coded for what these periods meant. So at the end of the second and the 3rd exposure, day 721, these are after stress exposures. So I'm showing this as the warm colors. Now day 14 is after the ambient recovery period, which I'm showing as a cool color here in blue. So first, for gene ontology enrichment, where we followed up by looking at specific functions, biological process molecular functions that were enriched. And here is showing persistently enriched terms. So in terms that were enriched throughout the experiment, regardless of the subsequent exposures. And the genes associated with these terms were attributed with histone methyltransferases and transcription factors in our conditioned animals, whereas the genes associated with enriched terms throughout the experiment were associated with fatty acid metabolism and glutathione components, or proteins that scavenge reactive oxygen species. Now, this can be potentially interpreted as a front-loading response because these were genes that were more abundant in the conditioned and naive animals throughout the experiment, and regardless of their subsequent stress encounters. So now for enriched gene ontology terms, molecular function biological process terms that are partially enriched on certain parts of the experiment. First showing the terms that were enriched on day 7 after the second exposure. And here we see the proteins in these enriched terms were associated with stress response. Mitogen activated protein kinase binding, regulation of apoptosis, and removal of repair of proteins. So we can summarize this as potential stress response to that second encounter. Next, during the recovery period, here's our terms that were enriched. Interestingly, for the conditioned animals, we see that they were enriched for several complexes of the electron transport chain during this recovery period, signifying that they potentially were performing upregulated energy metabolism. Whereas with the naive animals, we see fatty acid metabolism, similarly to what we found enriched throughout. We also see proteins involved with cellular quality control. And lastly, we have one last category that we found here, were enriched terms that occurred on both day 7 and day 21. I'm calling these as rapidly inducible because they occurred during stress, specifically during stress. And these terms, this specific category, only occurred with the conditioning effect modules. And what we found here was NF-kappa B activation, which is for innate immune response. We also see regulation of apoptosis and repair of proteins. So we may be able to summarize this as these rapidly induced proteins are associated with innate immune response and controlling cellular quality, or homeostasis of cellular quality control. Now, this may represent a fine-tuned response, where during the repeat stress exposures, and seemingly inducible because these terms were overlapped between the second and the third exposure, we see this increased expression for immune and stress-related proteins. Whereas during the ambient recovery period, when stress was relieved, we see a potential increase in energy metabolism due to an upregulation of genes associated with electron transport chain of the mitochondria. Now for our pathway analysis, lastly for pathway analysis. Here's showing day 7, day 14, and 21 for the conditioned animals and naive animals with genes that were more highly expressed by these two phenotypes. Just to guide your eye here, the main result that we see with the conditioning effect, we see a more variety of pathways that were enriched that included biosynthesis of amino acids, carbohydrate metabolism, and mitochondrial homeostasis. Whereas with the naive animals, we see this persistent enrichment throughout the experiment, regardless of the environmental changes that they faced. And they were associated with fatty acid metabolism, fatty acid degradation, and peroxisome activity. So here we see potential additional support for a fine-tuned expression by this conditioning phenotype, where we see a variety of pathways that were enriched. Whereas for the naive animals, we see a potential metabolic shift when faced with stress, in which we see elevated use of lipid storages by seeing these fatty acid metabolism markers throughout the experiment. So to summarize this last chapter, we had this obvious phenotype from chapter 2. We wanted to follow up to see what's going on the molecular level. For the naive animals, we found this continuous metabolic shift. We found these markers for fatty acid metabolism throughout the experiment. We also found continuous gene ontology enrichment, telling us that there was potential oxidative stress. As for the conditioned animals, we found fine-tuned and front-loaded expression, where we found that low pH encounters cause an increased abundance of immune and stress-related proteins, whereas during recovery phase, we see this increase in energy metabolism. We also found evidence of front-loading, where we see transcriptional regulators were more abundant throughout the experiment, and they were coded for histone methyltransferases and several transcription factors throughout the experiment. Now, to synthesize all these chapters together for everybody, main question of this dissertation was, what are the underlying mechanisms of the conditioning response? We first assessed what's going on during earlier life stages, where we found prevalent mortality, but this was not surprising because these stages may not face this pH condition in their natural environment. So we focused on the pedovelture and juvenile stages. So we did this first experiment where we wanted to see how they respond to repeat stress and what the carryover effects of this repeat stress. And we found evidence that earlier stress may precede growth compensation and metabolic recovery. For our second experiment, I wanted to see what does conditioning earlier in life yield distinct patterns in response during repeat stress encounters. And here we conditioned the pedovelture stage immediate post-settlement to yield naive and conditioned animals to see how they respond to repeat stress. And we found a distinct phenotype associated with decreased size and increased antioxidants by animals that did not experience conditioning, whereas animals that were conditioned showed increased performance and decreased cellular stress response. We then followed up with this distinct phenotype, this potential phenotype, to interrogate the molecular level, what may be attributed with this distinct phenotype. And for the naive animals under an acute exposure to hypercapnic seawater, when they did not experience this earlier in life, we see evidence of an increased transcriptional demand associated with beta oxidation and fatty acid metabolism. And fatty acid metabolism does yield more ATP than carbohydrate metabolism, potentially feeding that higher energy supply back to transcriptional demand. We also found that the transcription activity of the gene expression was associated with antioxidants. So this potentially tells us that the naive animals under low pH or hypercapnic seawater had a compromised, what may have produced high reactive oxygen species. And interestingly, beta oxidation and fatty acid metabolism does produce free radicals. So there's a potential negative feedback loop in which naive exposure to stress may increase free radicals and also increase the need for fatty acid metabolism, further increasing this pool of reactive oxygen species and causing stress. However, this is an area for future research. We did not quantify reactive oxygen species, nor did we quantify assays for damage. So further research is needed to really disentangle the role of reactive oxygen species in this effect. As for the conditioned animals, we found improved performance after they were conditioned and repeatedly exposed. This was attributed with, again, increased growth, respiration rate, and tissue biomass, and decreased cellular stress response. And we also found with our gene expression patterns, we see evidence of fine-tuned expression and front-loaded expression. And this may contribute to a memory of that previous pH encounter, previous stress encounter. We found some evidence with the front-loading expression for modifying chromatin status, where we saw histomethyltransferases were continuously expressed at a higher rate throughout the repeat encounters. So there's potential here to expand upon chromatin modifications, and a growing body of literature supports that both genetic and non-genetic interplay can affect gene expression patterns. So this box represents areas for future research. But incentivized from our as a jumping point from what we found with our phenotype here. So our main conclusions, we found that stress conditioning post-settlement may elicit beneficial phenotypic variation in Pacific geoduck. And this beneficial phenotypic variation was attributed with enhanced performance, as shown by increased size, respiration rate, tissue biomass, and also may fine-tune expression and front-load expression during repeated stress encounters faced later in life. Now let's translate this for aquaculture, where stress conditioning might be a viable strategy for aquaculture improvement. Currently, aquaculture uses a bioremediation strategy by adding sodium bicarbonate to their incoming seawater in order to increase the pH and allow the animals to develop normally. However, I propose that this is only beneficial in the short term and may leave the animals whose artificially propagated seed, unprepared for the grow-out condition that they will inevitably face later in life. Whereas stress conditioning may elicit mechanisms that may carry over during later life encounters when they will face these low pH seawaters in the dynamic systems during grow-out. But for future directions, we need some long-term tracking of these phenotypes to determine transient and both long-term implications of the early life exposure. And further research should also interrogate more omics mechanisms to determine whether there's genetic and non-genetic interplay affecting gene expression patterns, and also whether this response that we're finding here is transferable among other species of economic importance. Great, thank you everybody for coming. There's a lot of people to thank here. Specifically, I want to thank Jamestown Slalom Tribe Here's a picture of a beautiful hatchery in Vernon, Washington, where all this research took place. Specifically, Kurt Grinnell, who was the CEO of Jamestown's column Shellfish, he recently passed, but I know that he would love to see this research. Matt Henderson and Josh Valley, they were my housemates, and also they trained me on everything I know about geoduck. PIs and co-PIs, Holly Fatima, of course, for taking me under your wing as your student. It was an honor to be your student all these years. Several PIs from the University of Washington, Stephen Roberts, Greg Valapalis, and Shelley Trigg, my committee members, and also everyone in the Putnam Lab. You're all very uplifting and supported throughout the years, and our funders from FFAR. And that's all I have for you today.